are back for hour number 2, which is part 3 of chapter 26, amino acids and proteins. Keep in mind the big thing today is test 3 over the previous 3 chapters and the homework, and coming up, carbohydrates, so stay tuned for that. Let's go to the sideboard here and look at what we need to do now on sequence determination. Now, what do we mean by sequence? We're kind of jumping ahead a little bit. The sequence here is the order of the amino acids. Remember I told you they're linear, right? It's just the amino acids stuck together in a long chain. So here's XXX, how many number of amino acids stuck together. But let's say you've got a new protein or a new peptide. How do you determine what you've got? So this is a problem that's answered with some chemical analysis and some reactions that we need to do. What's the order, and what are those amino acids in that order? If a new protein is determined, this is what the approach is here. The first thing to do, and a lot of times these 5 things are used in combination. We don't use just one thing to determine the whole sequence, especially if we have hundreds or thousands of amino acids to figure it out. What's normally done is a partial hydrolysis of the pure protein, and that will give fragments, smaller fragments. And these fragments then are easier to determine. And how do we do it? Partial hydrolysis. We treat it with acid and heat it up. So we begin to break those amide bonds, okay? Another way to do it is to use specific proteases or peptidases. And these will what? These will begin to hydrolyze at specific positions. So this is on the C-terminal side of what? Lysine and arginine. So those 2 amino acids on the C-terminal side with the trypsin. Chymotrypsin is on the C-terminal side of the aromatic amino acids. So phenylalanine, tyrosine, and tryptophan, the big ones, the big aromatic ones. And that's again on the C-terminal side. So that will give specific smaller peptides or smaller long chains, but we'll know the spots where those come off, okay? And these peptidases, they're just like reagents. They will take the protein and then break the chain at these specific sites. So trypsin and chymotrypsin, you can just say trip, okay, if you want, on an arrow, or chymotrip. There's also carboxypeptidase. Carboxypeptidase will cleave on the C-terminal side of the end amino acid. It'll just knock off the end one. So yeah, carboxypeptidase. And there are other specific proteases, but these are the 3 we expect you to know. And then there's the Sanger determination. This is for the amino acid on the N-terminal side. And that amino acid will actually do a substitution, nucleophilic aromatic substitution, an NAS type reaction. On this reagent, this is what? Dinitrofluorobenzene. And it has to be this arrangement, the 2,4-dinitrofluorobenzene. Why? Because if you push that in as a nucleophile, you have negative charge here, here, and here, and that's stabilized by those nitro groups. We've seen nucleophilic aromatic substitution before. And then when this is digested further with acid or one of the proteases, then you get that amino acid on the end here that has that nitrogen with this aromatic group, this dinitro-aromatic group, and that will have a different appearance in the UV spectrometer, or it can be determined by mass spec where that N-terminal group was. That's called the Sanger method. I'll show you more details of the Sanger and the Edmond here, which is another reagent. This is a phenylthiol isothiazole reagent. And this group will form a hydantoin. You'll see that in a second here. And that will actually form a heterocycle off the N-terminus each time. And this can happen sequential. So after you take off one N-terminal amino acid, you can put in more of the phenylisothiocyanate and get more hydantoin to come off. And each hydantoin corresponds to one of the specific amino acids. And then by mass spec, you can determine which amino acid was at which spot within your peptide or your protein. So the Edmond degradation is actually probably the more important one, along with the peptidases, as I The Sanger is kind of archaic. It's just for one shot, the one time on the N-terminus. The partial hydrolysis just gives a mixture, kind of a shotgun approach to hydrolyzing things. But that often gives these fragments that are easier than to sequence using trypsin, chymotrypsin, or the Edmond, okay? So that's why I said a lot of times, well, almost always, they're used in combination. Mass spec is a more modern approach, especially multi-dimensional mass spec, where you have these peptide fragments, you form the molecular ion, and then these things fragment and give fragments. Those fragments can be analyzed in the spectrometer, and then you can further see how those fragmented polypeptides fragment into the individual amino acids. And it's very sensitive. You can easily detect different amino acids, even with very small amounts of the original protein. When we've talked about mass spec before, remember that's a spectrometry where you form the ion, then you collect those ions and you count how much there are, and you get the molecular weight, the mass. And of course, each amino acid, except the isomers, leucine, isoleucine, are the same. You have to do a little bit more there. There's some great differences that you can differentiate those, but you get the amino acids, the resolution there you need. And again, what the order is, that's the whole goal of this. So let's look up on the board here and see what we need to do there for the sequence analysis for the Sanger determination. So here it is, the fluorodinitrobenzene, and here's a peptide here, VEGRL. And if we treat that C at the right pH, we can have this ionized here. And what? That amine is nucleophilic. The arginine one is there, the guanidinium, but that's more basic. We can find a pH where we keep that one protonated. The free amine's the better nucleophile. If it adds here, you see it'll covalently link to that aromatic group. And then if you hydrolyze this here, you can see there's only one amino acid that has that aromatic group. That'll change the mass of that group by mass spec, you can see that. Or by UV spectrometer, it actually shows up as a yellow compound. You actually use your naked eye to figure that out too. But anyway, the Sanger's limited to just the N-terminus characterization. Then we got the Edmond, a little bit more here. So here we got the phenyl isothiocyanate. That's this new functional group, you could say here. The carbon in the middle is electrophilic. That's where the reaction is going to start. And here's that same peptide, right pH. That's nucleophilic to add to that carbon. So there's some proton transfer steps. I'm not showing the whole mechanism there. That's the thiourea intermediate. This thiourea thing here, you see, can undergo a substitution here onto the carbonyl of the amide, holding this N-terminal amino acid on there. So this valine, it can undergo this nucleophilic attack by the sulfur here. Got the lone pairs donating in there. Gives this intermediate. And then what? Loss of water there can give you this breaking down. This is a tetrahedral intermediate, right? That's the amide. And if we push the electrons back down and then kick this off and then protonate that nitrogen, look, we've just pulled off that amino acid as the thiazolone intermediate. You'll see more there. But we can take then EGRL and do more cycles here. That has a new N-terminus. So within the Edmond degradation, we can actually automate this and add in phenyl isothiocyanate for a given amount of time to bring off one of these thiazolones, which rearranges, by the way, to the hydantoin, which is right here. Now, you can go through those steps mechanistically, and I think there's some homework where you can look at those mechanisms to rearrange the thiazolone into the phenylhydantoin. But the first one to come off is this one right here, which has the isopropyl there. So that's what amino acid? That's valine, okay, coming off there. So the next one to come off will be this one, which is the hydantoin that corresponds to what? Glutamic acid. That has that side chain there. That has a different mass in the mass spec. You can determine that one. Next one to come off is this one. Well, that's the simplest style hydantoin, and that's from glycine. Then the next one coming off is the arginine one, right there, or the guanidinium. And then the last one to come off is right there, the leucine one. So the sequence can be determined directly from the Edmond degradation. Now you have to have smaller peptides to be able to do that. The sensitivity drops off if it goes through too many cycles. But you can do up to 50 mers or so with Edmond degradation, looking at each phenylthiohydantoin heterocycle coming off into the mass spec. Here's partial hydrolysis. We need to talk about this, and we'll go to the board in a second for this. If you chopped up that same peptide with 6 molar HCl, that's very strong. That's what, pH negative 1.5 something? Very strong acid. So you'll begin to hydrolyze these amide bonds here. But you can do it for a limited amount of time and just get these fragments, okay? So you can get val glue gly, which is this fragment right here, hydrolyzing that one. Or you can hydrolyze here and get this one off, and then that one off, and get this one. So all these different fragments can come off. But their sequence is maintained in these smaller fragments. And that's what I want to show you. We didn't completely hydrolyze all the way down to the individual amino acids. We partially hydrolyze, and we maintain some of the sequence identity within these fragments. And that's kind of the key thing here. We're going to have to look at that. And so if you could sequence those little fragments, then you could line them up, and there are computer programs that help you do this. They slide along and see where the sequences line up. Then you can propose a structure for the actual beginning pentapeptide you see here with the 5 amino acids. So let's go back to the board here and go through an exercise where we do this together. And I'll give you a little bit of time and we'll work on this. So let's see. Let's say we've got a 14 mer, okay? Now what does that mean? We've got a peptide that has 14 amino acids in it. And we're going to take first, we're going to take chymotrypsin, okay? Chymotrypsin, you know, in regular water, and that's the amazing thing. Chymotrypsin hydrolyzes these amide bonds under neutral pH. And trypsin, chymotrypsin, they do this quickly too. And that's an amazing thing. Peptides are indefinitely stable in neutral water, but when we add the protease in, it begins to chop them up. We'll have to look at that mechanism. And the fragments you get here are AKF, and you get GDW, and you get SRMBRY, and you get LH. Okay? Now, these sequences then are known, and we get these from the Edmond degradation. Okay, so our original 14 mer is chopped up or digested with chymo to give these 4 new guys. And chymo, remember, hydrolyzes on the C-terminus of the large aromatic one. So phenylalanine right there. Oh, there's tryptophan right there. Here's tyrosine right there. And look, we can kind of tell what's on the C-terminus now, because that's histidine. That's not a spot where chymotrypsin cleaves. So is that the C-terminus? Maybe. So we're already learning quite a bit about this 14 mer. We know these subsequences are within the 14 sequences, and we have an idea of the C-terminus. So you're kind of like a detective now. You're adding up some clues about, and you're going to put it together to propose then the most logical sequence for the 14 mer. Our next reaction is to take the 14 mer with trypsin, so then we get AK, and we get MVR, and we get F, G, D, W, S, R, and we get Y, L, H. And there's H again. There's Y, L, H, right? And that's our C-terminus, maybe? Well, it's at least still consistent with that. And where does trypsin cleave? Trypsin cleaves what? Next to lysine, yeah, K. Arginine, yeah, and arginine. Okay, so that's consistent with that. And here's our, you know, YLH right there. Okay, so this is linked up with some of those spots, and you can already begin to see some overlap there, right? AKF, and here's AK, right? Here's W. Where's W down here? Well, it's right there, right? And DW right there. And let's see, SR is right there, and MVR right there. So you can already begin to see how things might be overlapping. In fact, we can begin to line things up now. So let's say we've got the alignment. Okay, so this is enough data, actually, to begin to figure out this 14 mer. So how are we going to do this? Well, we've got AKF, okay, AKF right there. And then we've got, let's see, MVR. I think I've got that one right. We've got a bigger piece, which was what? SR, MVR, Y, right? It's lining up right there. So those fragments all line up. MVR, and then we've got SR right here, right? That's R, and there was a W before that, DGF, okay, lining up right there. So you can kind of see how those line up, right? And then let's see at the end, we've got LH, and we've got the Y right there, okay? So can we propose a structure? Let's see. So what would be the proposed structure here? So all of these lining up, AKF, and then F, you know, on, where's this other F line up here? F right here, okay. This one lining up right there with the F right there. F, where was it right here? FG, yeah, FG, okay. DW, yep, R right there. And let's see the Y. Ah, the Y lining up right here. Y, LH. So is that our 14-mer? Let's draw it out. Let's propose a structure here. Is it A, K, F, G, D, W, S, R, M, V, R, Y, L, H. And again, this is our C terminus, and this is our N terminus. And all this data is consistent with the chymotrypsin and the trypsin digest. We saw all those fragments there. And this is it, right? 1, 2, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14 more. Yeah, there it is. And that's a complicated one. Okay, I'm kind of jumping ahead here. There's some simpler ones you can do homework-wise, but this gives you an idea of how it lines up. So the one I showed you on the overhead was just partial hydrolysis with acid. But now we're using trypsin and chymotrypsin to cleave at these specific spots. So let's kind of review here. Let's see, chymotrypsin cleaved what? Right here. Phenylalanine and W, there it is right there. And what? Tyrosine. Okay, so that was chymo. Okay. And where did trypsin cleave? Trypsin cleaves at arginine and lysine. So there's a lysine, there's an arginine right there. Arginine right there, yeah. So within that, we get those fragments that then we can overlap and begin to figure out there. Okay, so very good. Let's go back up to the overhead here. Sorry. And see what we need to do. Oh, this is just a summary of trypsin, chymotrypsin, and carboxypeptidase. We could have added that in, and I've already summarized that. I think we're fine there. Now, let's get into the structure of these things. So, if these things get big enough, they're going to fold up and form these different macromolecular structures, right? We're going to need to talk about these individual structural elements. The primary structure, secondary, tertiary, and quaternary structure. Here's Linus Pauling, the famous chemist who came up with the electronegativity, the structure of the amide bond, and the alpha helix. He almost had the correct structure for DNA, but Watson and Crick beat him to it. But he's a famous American chemist at Caltech. Probably heard a little about him. And also the vitamin C guy. We're going to see a vitamin C issue. So there's a couple issues related to Linus Pauling here. He won 2 Nobel Prizes, one for chemistry and one for peace. He's kind of a unique person there, winning 2 Nobel Prizes in different areas. Peace was for the nuclear armament détente issues with the old Soviet Union and America. He organized a number of Soviet and American scientists to talk about peace of nuclear weapons. Here's the structure of a peptide chain. All sorts of R groups, all sorts of conformational freedom. So each one of these blue rotations here can occur. So there's an infinite number of conformations here when these things begin to fold up. But luckily, we have not only this primary sequence, which is just the order of the amino acids, we also have secondary structure domains, which are the alpha helices, beta sheets, and turns. And then how these things fold up, that will be the tertiary thing. And then if there are multiple chains, that will be the quaternary thing. How is this done? Well, when a new protein comes about, we overexpress it and purify it and crystallize it. Here's a crystallized protein, kind of a pretty looking structure there, seen under a microscope. Then we bombard it with X-rays and collect all the diffraction data. That's what graduate students are for. They do all the work, right? The professor, the boss, gets all the glory. Students do all the work. Oh, but look, she's smiling. So her crystal diffracted well. She uses the Bragg equations with the computer, of course, to generate then the 3D coordinates for every atom within the molecule. And then the computer builds the actual structure. Okay? By then, you already know the sequence, the ordering of the amino acids. Here, we need to know by X-ray crystallography the secondary and the tertiary structure, which are these folded up proteins here. How many are there in the human genome? Let's figure out the numbers here. Well, there are estimated to be between 23 and 25,000 proteins that are produced in the human cell. Those are genes that correspond to proteins that are made. We only know the structures of about 3,000 to 4,000 of them. In fact, all the other ones, we really don't know a lot about their function within cells. So there's a lot of opportunities to win more Nobel Prizes. We'd like to have a computer program that could actually go from the primary sequence and go directly to the tertiary structure. We don't have that yet because there's an infinite number of conformations that can occur here. So it's very hard computer-wise to just go from the sequence directly to this. But we do know some amino acids that tend to be in helices and some that tend to be in beta sheets and some that tend to be in turn regions. So I'll mention some of that, but it's way beyond the scope of our course to talk about all these things. Let's go back to the board now and talk about the protein structure and these different topics. So what do we need to know here? Protein structure. So now we've got, more than 50, we might have hundreds or even thousands of amino acids in the chain. The structure of proteins breaks down into 4 areas. The primary structure is what? The sequence. We've been talking about sequence, and this is just the order of the amino acids. From the N terminus to the C terminus, okay? Where they all are. And it's very important to keep that orientation straight. N terminus on the left, C terminus on the right, and where they are position-wise within that sequence, okay? That's called the primary sequence. Everything else is a consequence of that primary sequence because it comes off the ribosome, we'll see later, as just this linear chain, right? So how do we get it folded up? How do we get the structure and function of all these proteins? Well, luckily there are localized domains here called secondary structure. These are the alpha helices, alpha helix, singular, which will be these corkscrew-shaped things, these coil shapes, and they're right-handed helices. The actual topology of the object has chirality in itself. And that's engendered because of the stereocenters of each amino acid being L. The L have the right-handed corkscrew helix there. But we also have beta sheets. These are the extended zigzag things that kind of bite back on each other, and we'll talk about how they're stabilized here. These are extended or open zigzag sheets. Okay, and we'll see how these are abbreviated there. But yeah, so those are the secondary structures. Also terns. The term regions connect the alpha helices and the beta sheets. They're not as defined. The helices and the sheets are very well defined, localized secondary structural elements. Okay? Tertiary structure is the fold into the three-dimensional object here. We have the globular ones, right? These globular ones tend to be the enzymes. They're involved in metabolism. These are the catalysts for different reactions, right? And we'll learn about quite a few of those. They're water soluble. They have all the polar and ionized ones on the surface area, out on and then the interiors where they have the non-polar groups. But these things ball up and form these globular shaped things. So how does the folding work with that? Well, this is also plus cofactors. So often a protein, especially an enzyme, will need a cofactor like a heme ring or NADH or FADH. We'll learn about some of these other cofactors. These bind non-covalently to the folded up protein, but they have to do with the function of these globular things. We also have the fiber proteins. These fibrous proteins like fibroin, silk, collagen, a lot of different fibrous proteins that hold together joints and skin tissue. And we also have keratin in hair and fingernails and things like that. And these tend to bundle up with each other. And that's kind of the next topic. If these things have multiple proteins linked together, protein will say X there. Most of these fibrous proteins are bundles there. And the fibrous proteins are what? Insoluble. Okay? So these will tend to be linear things that are kind of wound around each other to form structural elements there. The key one there, I think, is collagen. And collagen is an important one. It has hydroxyproline in it. And hydroxyproline is a post-translational modification where it puts an extra hydroxyl group on proline. And that allows for more hydrogen bonding and more strong fibers, collagen here. Now, how does proline go to hydroxyproline? Maybe you've heard about that, right? Vitamin what? Vitamin C. Now, if vitamin C is not in your diet from fresh fruits and vegetables, guess what? You don't make hydroxyproline and your collagen's not very strong. And you get a disease called what? Scurvy. That's right. It's a historical disease that sailors often suffered from this. The British finally figured this out. That's why they're called limeys. They're sailors historically, and that's because they started taking fresh limes and oranges with them on their sea voyages that would go on forever. And they had still maintained their collagen. If sailors don't eat fresh fruits and vegetables, just like us, they'll have all sorts of disease conditions. I'll show you that. That has to do with the fibrous protein collagen, which actually bundles up. So there's multiple chains that come together to form these strong fibers of collagen. And that's an issue of what? A quaternary domain. Same thing here, we'll learn a lot about enzymes here. We'll learn about myoglobin and hemoglobin. Hemoglobin has alpha and beta chains. But when the two alpha chains plus two beta chains come together, then that's a multiple complex, a quaternary structure. And that's the actual thing that grabs on the oxygen in the body and the lungs and delivers that oxygen to the peripheral tissues. So there's a couple things you've probably heard about there already. We'll look at the structure of myoglobin here pretty carefully, and we'll illustrate all these structural elements here. I think that's enough on the board there. Let's go back up here. Sorry, we're giving the camera a little workout here. And again, you have these graphics on Learning Suite, so check them out there. So there's the issue of proteins folding up. We got the localized domains, a couple of them there, the alpha helix and the beta sheet. We'll look at them a little more closely, and then we have examples of the tertiary fold here. Here's insulin, which is kind of at the boundary between peptide and enzyme. Insulin is a hormone, okay? So it's not a catalyst here. And it's two smaller chains, 30 and 21, so it's about 50 or so, held together with disulfides here. You can see the disulfides in yellow here, the two sulfurs coming off the cysteines. And you already see these abbreviated spiral structures here, these corkscrew structures. What's that? That's an alpha helix right here, right? And another helix here, another helix here, and some of the side chains illustrated. So there's a lot of ways to represent protein structures, these cartoon representations. And why do we use those? Well, we've just got too darn many amino acids and atoms to keep track of. But you can see some here. I left behind some of the side chains. You see what? A benzene ring and this methylene. So that's what? Phenylalanine? No. There's a hydroxyl right there. So what amino acid is that coming off? That's tyrosine, right? So you can see that. Yeah, you can identify a couple others there, which is kind of cool. So there's different ways to represent it, but that's insulin. That's the hormone that regulates the glucose intake from the blood into the individual cells. Insulin signals the glucose receptor to bind glucose and to bring it into cells, right? You've heard about that. If you're deficient in insulin, you have what disease? Right, diabetes. There's the abbreviation of the individual side amino acids and the two chains going again from the N terminus to the C terminus, there and there, and then the linkage between cysteines. So that's another tertiary stabilization effect. Okay, so it's the alpha helices, beta sheets, and the disulfides are part of the tertiary. I'm sorry, the tertiary folded one is where the disulfides are in. The alpha helices and beta sheets are secondary domain elements, right? Okay, so here's the problem, right? Here's an enzyme, tryptophan synthetase A. It's a protein, and what does it make? It makes tryptophan. So a lot of these proteins are named in a straightforward way that describes something about what they do. But here's the problem. It's 267 amino acids, and here it is written out in its IUPAC name, which is the prefix starting at the N terminus, methionine, glutamyl, arginyl, tyrosine, glutamyl, serial, leucine, phenylalanine, alanyl, so phenylalanine is for phenylalanine, right? And you go all the way here with these YL names for prefix, and all the way at the end is serine at the C terminus. So yeah, the IUPAC name becomes onerous, it's too big, and so these common names right here are also accepted by the IUPAC. Tryptophan synthetase A, okay? 2,000 letters in the name of the enzyme, but for 267 amino acids. But that order is important. You can pick out the order and go back to the one-letter code. Anyway, here's the secondary domain elements, the alpha helices, drawn out with all the atoms. You can see some specific ones. There's tryptophan, right, with the indole. There's a methyl sulfide, that's methionine. Here's a benzene with nothing on it, so that must be phenylalanine. So yeah, you can pick out a lot of them. And a lot of times we strip off all the side chains, and that's what you see here. You just see the backbone, the polyamide. So that's like polyglycine, okay? And you can add on all the side chains depending on what the sequence is. And then also, what holds together an alpha helix? Well, yeah, these hydrogen bonds that can occur between the NH group and the carbonyl. Each green dot here, dotted line, is a hydrogen bond. It's every third and a half amino acid up, forms an intramolecular hydrogen bond. And they're weak on their own, but if you add up a bunch of hydrogen bonds, that begins to stabilize that. The amino acids that tend to be in alpha helices are methionine, alanine, leucine, glutamic acid, and lysine. Okay, malic. That's how I remember it. I don't expect you to memorize that. I just want you to know that there are some amino acids that tend to fit better in alpha helices. And it's kind of a steric thing, right? It's coming off this corkscrew backbone, and they fit better in that arrangement there. So that's how we strip it down to see it. Here's how it's often abbreviated, different structures you've probably seen in different books. It's just these ribbon cartoon diagrams. So this is a corkscrew or the coil structure here. Doesn't tell you anything about the amino acids involved, but it tells you that this is in a conformation that has this right-handed staircase helix arrangement there going up in the right-handed way. The topology of the twist is chiral itself, right? It's a right-handed screw topology. And then this part up here is undefined. This is a turn region where it's not well defined. Okay. Then we got the sheets. Beta pleated sheets are sometimes called in reference to pleats in clothing, I think. Pleated skirts and pants, I think. Pleated pants, I don't know. Craziness. But here's the extended zigzag conformation that we've been drawing on the board for peptides. And if we have them anti-parallel, here's C to N, and then N to C, we have these hydrogen bonds that can occur across two of the extended zigzags. So this is an anti-parallel beta pleated sheet right here. And then there's also parallel sheets. These are a little less common. Why do you think they're less common? Because look, the orientation of the hydrogen bonds is a little bit off center. They're a little bit less stable than these, which are right dead across. Okay, these are more stabilizing, these hydrogen bonds. And the ones, amino acids are shown here that tend to be found in beta sheets. And here's the representations of these. So here's a beta sheet and a turn region. Turns, that's where glycine and proline usually hang out. There are all sorts of repeated motifs that can be seen within protein structures. These are the so-called beta barrel arrangements. If you have a bunch of these pleated sheets and they kind of fold around and form a barrel there. Here they kind of twist past each other, but there's still extended zigzag orientation with hydrogen bond. Here's a anti-parallel one, which is kind of rare. And here's the regular parallel one right there. Okay, so yeah, it's the sequence, that's the primary structure. These secondary domains, alpha helices, beta sheets. And then the 3D, where it folds up, forms a globular thing or the fiber. And you have to add in the disulfides and the cofactors at that point, right? That's part of the tertiary thing. Then if you have multiple chains, this will be typical of hemoglobin that we'll see here in a second. Will be multiple coming together. Here's a nice graphic that shows all of the interactions that are possible in proteins. Hydrogen bonds are very common. Here's a sheet, and here's an alpha helix with the hydrogen bonds, the dotted red there. We also have hydrophobic interactions, the nonpolar things, which will tend to be in the interior. So here we have valine and isoleucine. Hydrogen bonds can occur across the chain right here. So these are in the main chain, carbonyls and NH. And we can have hydrogen bond between a side chain and the main chain, like tyrosine to a carbonyl, a banana. Here we have a salt bridge. That we have an interaction there. This salt interaction is ionic interaction. Here we have a polar interaction that's hydrogen bonding. And here we have a hydrophobic interaction also right there. And here's our disulfide, which is also the secondary interaction. Here's some fibrous proteins. I mentioned a couple of these. Collagen shown here. Collagen are these bundles of polypeptide chains here. And we have proline, and we have hydroxyproline. Hydroxyproline from vitamin C gets that extra hydrogen bonding to the structure. Now we have keratin in hair and fingernails. They also form these alpha helix bundles. Okay, yeah, you see the red bundles there, so it's similar to collagen, I'd say. Silk is a little bit different. Silk is a bunch of beta sheets stuck together, which is kind of strange. And that's with glycine and alanine. This is the structure that inspired Wallace Carruthers to do nylon with the polyamide structures. These things can bundle up, so this is a tertiary structure. This is a so-called beta, or an alpha helix barrel type structure with a bunch of them there. And they can come together and bury their nonpolar groups in the interior and have the polar groups on the exterior. And you've probably heard about membrane-bound proteins that can span a lipid bilayer membrane. The amino acids in the interior here point toward the lipophilic, the hydrocarbon, the long fatty acid chains here, which are nonpolar. So nonpolar amino acids will tend to be there. And a lot of these have channels. They can open up and let molecules come through here. A lot of membrane channels, like the glucose channel. They can have a ligand that changes the conformation and then changes the activity of the protein based on that structure. All right, myoglobin. We're going to spend a little bit of time here for the last part of the hour and go through a little bit on myoglobin. So just know the basics of it, right? It's a big protein. Here's the sequence from the end of the C-terminus, transports oxygen, and it has a cofactor, which is the heme ring right there. Folds up, and what do you see here? A lot of alpha helices and a lot of loop regions too. So those are undefined secondary structure. And then hemoglobin has a quaternary structure. It has two alpha sheets, which are shown here, alpha proteins, which are shown there. A lot of alpha helices, and then two beta proteins in orange here. And there's actually 4 heme rings that bind oxygen there. And there's a lot of structural information here. You'll learn about biochemistry, how its conformation changes the affinity for oxygen, which is kind of cool there. There's a histidine, imidazole. That amino acid side chain sticks right over the iron and helps regulate how the iron binds oxygen, which is kind of cool there. Sickle cell anemia. Well, you've probably heard of that disease. This is where erythrocytes, or red blood cells, form these sickle structures. And why is that a problem? Well, they're more rigid and they don't fit through the microcapillaries in the body very well. They restrict circulation. And this sickling is due to just one single mutation on hemoglobin. We'll have to look at that. These are normal erythrocytes. Here's one that's beginning to sickle. Most of them look like these things here. By the way, the red color is due to the iron thing there. It is packed full of hemoglobin. So these are actually cells with lipid bilayer membranes. But when the mutation occurs, they don't bind oxygen as well. They're not as red, and the hemoglobin begins to oligomerize. It forms an insoluble fibrous structure that forces the cell to sickle or form this elongated, kind of pointed type structure here. And let's look at the structure here a little bit on what the heme looks like. Yeah, it's an iron polyheterocyclic ring here. The nitrogen lone pair is pointing right at the iron. When it binds oxygen, it goes to the relaxed state, which is down here. If it loses oxygen, it's so-called tense state. It changes the conformation. We'll learn a little more about that later. It has to do with slight changes in the pH, protonating some of the side chains. And that's one thing we need to talk about. If you change the conditions under which a folded up protein is under, it can denature, it can change its folding state. What things might change that or denature a protein? Yeah, if you change the pH, you're going to protonate or deprotonate certain side chains. And if you add heat, you can change it from a globular structure into a fibrous one. Okay? And that's the cooking process does that with a lot of different proteins and different foods, changes the structure. It still has the same primary sequence and a lot of the same secondary sequence elements, but heat and pH can change them. Also heavy metals, lead and cadmium and metals such as that can change the folded state of protein. Some of that denaturing can be reversed back to the native folded state, but other states will stay irreversible in one new folded state. But here's subtle folded state changes based on oxygen being present within hemoglobin. Some proteins undergo dramatic changes in their folding, like these beta amyloid proteins in the brain. If they begin to aggregate, you form these fibrous plaques within the brain that can lead to different diseases there. Alzheimer's disease is probably the most well determined one for that. And that's simply catalyzed by some of these aggregated different foldings here. Here you go from the alpha helices to the extended beta sheets. And these sheets things can actually catalyze the refolding of the individual proteins there. So yeah, that's kind of a crazy disease there. It's not based on a virus or a protein or a mutation in DNA or RNA. It's based on a refolding of a protein. So yeah, that's Alzheimer's. We'll get into this in a second here. I'd like to back up and look at Proteopedia again here. We've got the graphic here. I think you can see that. I've shown you Proteopedia before. You just go to the main table here, main page, and you can look at basics of protein structure. And one of the key ones they already have linked up here is myoglobin. Myoglobin's in muscle tissue and heart tissue, and also aquatic mammals. In the light blue here, you just see the main carbon, alpha carbons, just linked together in chains. You can already kind of see helical regions there. And what the cofactor, the hemoglobin, shown right there. So let's highlight the heme ring. And there's a number of these. The Proteopedia is designed for academic applications, both in class and students on their own. So feel free to review these concepts going to Proteopedia and clicking through these. All of the green buttons let you change the image there. And you have control over the image. So now it's highlighting in solid balls here, space-filling balls, the heme ring. And it can further highlight the iron atom. It's the brown atom right there. And yeah, you can put it on spin maybe. Let's see if it'll stay on spin. Yeah, there you can see it. Or manipulate with your mouse like I'm doing now. Okay. Let's see, ball and stick model. This one gets more complicated. That adds all the atoms now. And actually each alpha helix is color-coded now. You can kind of see the helices. I'll highlight those again in a second there. But yeah, that's the problem with proteins, just too many darn atoms, right? We've got to have these abbreviated cartoon-type structures being to keep track of them. But that has all the atoms, including the heme ring right there, the darker structure. Right? And then we've got the hide the heme group. Yeah, get rid of that. We'll just look at the protein now. Okay, so it disappeared there. Polypeptide chain. Let's just look at that. Yeah, so that's highlighted out. Now, amino acids with positive charge. I'm clicking on that next button. What are the amino acids with positive charge? Okay, right. They're the ones that can be protonated, the basic side chains. So what are we talking about? Lysine and arginine. And here they are in red, and they're on the periphery. They're on the surface of the folded up protein. Why? Because they're polar. They want to partition toward water. They want to be solvent because they're ionic, right? They're charged. So yeah, that's where they tend to reside. And that's a factor of what? Folding up a protein. Let's look at the negative charge ones now. I'm clicking on the next one there. Yeah, they're the blue ones. What are the negative charge ones? They're the ones that are acidic. So we're talking about aspartic acid and what? Glutamic acid. And they're on the surface, okay, along with the other charged ones. And how about the polar amino acids? Where are they going to be found? Glutamine, asparagine, serine, threonine, tyrosine, yeah, the green ones. And you know, this is a space-filling model. It's kind of hard to pick out the individual amino acids. I'll go back and hopefully we'll be able to see some individual ones and where they're at. But you see the interior is kind of hollow right now. And that's because that's filled by what? The non-polar amino acids. Clicking on the next one. Ah, it's all the purple ones. There you go. So yeah, Proteopedia is helping us out here. All those purple ones are where what? Glycine, alanine, valine, leucine, isoleucine, phenylalanine are all held in the interior. Let's go back to the ball and stick model. There it is. You can begin to see some of these there. Here's a blue charged one there. That would be what? That would be lysine. You can kind of see it there. But yeah, and then the non-polar ones are the purple. You see that they're heavily dominant in the interior. Now that's not always the case. Every now and then you see a polar one in the interior if it's part of the active site of an enzyme or other things. Anyway, but I think you get the key there. Alpha helices. Let's highlight that. Yeah, back to the cartoon. And then the turns are undefined. They're kind of loop regions, we call them. Yeah, they're the darker black lines. Okay, I think that's enough on that. Let's go back to this. Hit the full view there. Okay, so what are we talking about here? We're talking about enzyme activity. Okay, an enzyme converts a substrate into something that's new here. So here we're cleaving this substrate, hydrolyzing an amide bond or an ester or whatever, breaking something off this substrate molecule. And here's the enzyme, the thing that actually catalyzes it or lowers the transition state and makes that reaction faster. And it's binding here, right? And so how does a substrate here, a small molecule, bind to a protein? Well, it's all the same interactions. Hydrogen bonding, the shape here, salt bridges, hydrophobic pockets there. It's shape complementarity and polarity complementarity. And then there's something in the active site that then affects the reaction. A lot of times it's a cofactor or some molecule there. So here's one, binding an active site here. This is so-called lock and key model where the conformation doesn't change. What we know there is both conformations now change quite a bit upon binding. That's the induced fit model for binding. We'll learn this later on. Here's sucrose going to glucose and fructose. This is a hydrolase enzyme using water to break this linkage here. The ether bond with what are acetals between the two sugars, and then adding water there to break it. That's the hydrolase family of enzymes. Sucrase is called. Sucrase, it lyses sucrose, which is a disaccharide. We'll learn that later. There are 6 classes of enzymes. Oxidoreductases, which do what? Oxidize and reduce. Here's ethanol going to ethanal. The aldehyde from the ethanol, yeah, it's an oxidation. And this alcohol dehydrogenase falls in this category. Know the basics of these classes. There's 6 of them. Transferases transfer a functional group from one thing to another. This is transferring an amino group to a ketone group. Okay? This alanine transfers, transaminases. Okay? And then we've got hydrolases, which use water to break bonds. So here's a peptide bond, and we're cleaving that. So these are the peptidases, trypsin, chymotrypsin. We've just been talking about that. We'll have to go back to the board in a second here and show a little bit more details on that mechanism. Then we got lyases, which break things off molecules without using water. So if you go from pyruvate to ethanol, that's eliminating CO2. That's what? Decarboxylase. So we're not using water here, but we are breaking bonds. So it's a lyase. It's breaking bonds. Isomerases, just what it means. It takes one isomer to another. So here's the cis isomer, malleate, to the trans isomer, fumarate. Okay? So there are other enzymes in this area. Some of the specific ones are shown there. Ligases, what do? They ligate or put things together. Ligatures are linkages between structures. So there's putting on CO2 back on a molecule. Yeah, so that's a ligase. And there are only those 6 classes, so it's relatively simple there. Let's look at the structure, the mechanism of the active site of trypsin. Trypsin cleaves on a carboxy side of what? Arginine and lysine. So here it is. Here's our peptide we're going to have to cleave. And black is our substrate. And the active site is a serine at 195 position. That's the order where it is in the enzyme. 57 is a histidine, and at 102 is a aspartate or aspartic acid. Okay? The histidine here can deprotonate the serine, and as it grabs that proton, liberates the electrons off serine to go in and attack the carbonyl of the amide. There it is, tetrahedral intermediate. Oh, we've seen that before many times. This is a derivative of carboxylic acid, right? Here's our tetrahedral intermediate. And this can break down, right? As these pair of electrons come back down, kicks off those electrons, and there's our free amine that can come off. Now this generates an ester. We'll have to see what ends up with that. That ester then can get hydrolyzed with water, and it could go back to serine and liberate what? The carboxylic acid here off the top part. So that's the basics of it. A lot of these hydrolases undergo these transitions where there's a tetrahedral intermediate. Some of these can be isolated and steady there. They're slow enough, but a lot of times they go really fast. Here's our carboxypeptidase, which has a serine in the, a zinc cation in the active site. It's a little more complex. It activates the water to attack the amide bond. And it leads to the cleavage of that N-terminal amino acid. So here's one coming off of peptide chain on the C-terminus, which would be phenylalanine. Anyway, there's a blow up of that zinc. The zinc acts as a Lewis acid to activate that for the hydrolysis. HIV protease, you've probably heard of some of the AIDS drugs to treat people that are infected with HIV, human immunodeficiency virus. These are proteases specific to the virus, and we've designed drugs that inhibit the protease, which is needed to form more of the virus. And here's leponavir, ritonavir. There's a bunch of these avir drugs that bind in the active site of a protein needed for HIV, the virus, to propagate. And here's a drug, cyclosporin, which binds 2 proteins, calcineurin and cyclophilin, which are needed to promote T cell proliferation. T cells are part of the immune system. If you can suppress that effect, you can then treat people who have organ transplants and they'll survive. Okay. All right, let's go back to the board here just to wrap up. And I wanted to show a little bit more of the mechanism of that protease effect. Let's see if I've got it. Where is it? It's got to be here somewhere. Maybe I don't have it. Let's see. Where is it? Okay. Ah, there it is. Back to the board. So, yeah, okay. Let's show just a little bit about this. Yeah, push the electrons. You got a little bit there. You got it in the graphics there, but I like to show it here. So here's the peptide bond, amide, between 2 amino acids R there, and we're going to hydrolyze this bond right here, right? And our enzyme is going to be this, which you're going to have a serine, primary alcohol right here. And then within the same enzyme, we have a base, which is histidine, imidazole, the lone pair. Okay? So I like to just show this. This is part of the enzyme. This is the catalyst. It's going to do the reaction. So we can take that proton off, liberate these electrons to go in and go up and form our tetrahedral intermediate. So let's see what that tetrahedral intermediate looks like. It's got serine there, okay, and the protonated base, and we've got the rest of the peptide or protein, right? Something like that. So that is indeed a tetrahedral intermediate, right? So what do we need to do now? We need to bring the electrons back down, and you know we can push this back off here to go back to the amide, but we can also push this off here, what, and protonate the nitrogen. And that's kind of the key thing, right? So if we bring this down, what do we have? We have an ester now with the enzyme, right? And we have the base back deprotonated, and we have the nitrogen of the cleaved peptide chain, right? So this is our substrate here, right? Here's our enzyme. And this is now what? An ester. Which is kind of an amazing thing, because we know how we can cleave esters with what? Water. So I won't show the rest of that, but let's just see that what's going on here. What goes back here is our substrate now, you see, has the free OH. Oh, and we left off an argon here. Sorry, there is that other side chain argon. The ester is a covalent link between the substrate and the enzyme. See that right there? And then we can hydrolyze that, right? By adding water, we can get back here on the substrate, the carboxylate cleaved there. We can get back the enzyme intact, right? And we've got the amino part of the substrate also right here. And depending on the pH, right? pH is neutral. These proteases, trypsin, chymotrypsin, work at normal pH, and that will protonate. I think that's the end of the story there. So that gets the cleaved thing. Cleaving this amide bond right there on the substrate. Both sides, you know, going on here. It's part of the chain there. The enzyme has the serine, which is the amino acid in the active site. This is the histidine imidazole, which takes that proton off and allows that to attack it. Now this alcohol on its own is not a good nucleophile, but it's the proximity of the specific base and the localization of that. There's also things in the enzyme that stabilize this O-. It's called an oxyanion hole. They're actually cation groups that point toward this O-, which helps stabilize that. The enzyme, you know, if it's not there, this amide bond is not breaking. But these factors can stabilize that. So it's the binding of it and then the proximity of these groups that allow us to break that from it. Anyway, that gives you the rest of the story a little bit beyond the book with the enzyme story there, but I think that gives a more complete story. And that, believe it or not, completes chapter 26 there. Next time we'll start on carbohydrates. Make sure you finish up test 3 tonight. And we'll see you next time. Very good.